am Mrs. Sloan, and this is part 2 of Honors Bio's Cellular Respiration discussion. So I first want to go over what we have talked about so far. So let me put myself, I don't know, somewhere over here. Okay, so last time we did an overview of cellular respiration, and we discussed in detail glycolysis. And we talked about the conversion of pyruvate to acetyl-CoA. We also talked about the structure of the mitochondria. So if this is unfamiliar to you, I would recommend that you watch video 1. We talked about our mules that we use to transfer electrons and hydrogens to our electron transport chain. And the 2 mules that we use are NAD and FAD, temporarily holding on to those electrons. We also discussed the 3 types of phosphorylations, of which 1 and 2 we are doing here in cellular respiration. Okay, so going back to here, I've already done glycolysis, 10 steps, 10 different enzymes, and you end with 2 pyruvic acids. In the transition reaction, you take your 2 pyruvic acids and, here's glycolysis, just a little quick review, okay? And then in the transition reaction, which occurs in the mitochondria, what you're doing is you're taking the 2 pyruvic acids, you're removing a carbon off each one, and once you have done that, you can also reduce a couple NAD, and now we're ready to go into step 3 of aerobic respiration, which occurs in the matrix of the mitochondria. Okay, and now I'm going to go ahead and put myself in presentation mode. All right, this isn't a pretty picture. I have a better one for you, but I want to show it to you just for a quick minute, and then I'll show you the better one that I have. Okay, first of all, you can see out here is glycolysis. If you can see my pointer, let me get a pointer. Un momento, pointer. Okay, so here you can see glycolysis, and then we do the preparatory reaction, move in, and in the center, Krebs, that's the name of the guy, citric acid cycle. Where it gets its name from is, remember those 2 acetyl-CoAs we just made through transition prep? Well, we're going to put one of those away, and we're going to take this one right here, this 2-carbon acetyl-CoA, and we're going to hook it up with a 4-carbon molecule, which makes a 6-carbon molecule, and that 6-carbon molecule is called citric acid, and that's where it gets its name from. Okay, so this is in your book, but I'm going to show you a simplified one. Okay, this is one that I made. All right, now, the 2 acetyl-CoA that we made from Krebs Cycle, we have 2 of them, we're going to put one away. This 2-carbon unit right here, and mind you, it has hydrogens and it has oxygens on it. The coenzyme gets kicked off as soon as we get in here. I am just following the carbons right here, so that's why it says C2, okay? There is a 4-carbon molecule that is preloaded in there. A 2-carbon connects to that 4-carbon and it makes a 6-carbon molecule. Okay, this is not glucose, it's just a different 6-carbon molecule. This is what we're going to do. It's kind of like we're doing this transition prep all over again, because look what we're doing. We're pulling off a CO2 and we're reducing an NAD. So what was once 6 is now 5, right? So now we're a 5-carbon molecule. What have we gotten out of the process? Well, we got another reduced NAD that we can cash out for ATP. Okay, now what? We're going to take this 5-carbon molecule, we're going to pull off another CO2, so it's going to get down to 4, and we're going to reduce another NAD that we can take to our electron transport chain. So now we have 2 of them, right? Then we have a phosphate group on this C4 molecule, so we are able to make a single ATP. This is through substrate level phosphorylation. It's a handoff, just like we did in glycolysis. Then we're going to pull some more electrons off and give them to our other mule, FAD. So here it is, the reduced FAD. So now we have 2 NAD and 1 FAD, right? Then we can pull off a few more electrons and reduce another NAD. So by doing 1 turn of the Krebs cycle, we get 3 NADs. And if you don't take in consideration the second law of thermodynamics, each one of these will net out an 3 ATP, okay? And because, stick with me here on the math, because we have 2 acetyl-CoAs, that means we can do this Krebs cycle 2 times, which means a total of 6 NADs if we do it twice. And each one of these gets you 3 ATP at the electron transport chain. So you could do the math on that. 6, right, times 3 is 18. So by doing the Krebs cycle, we get 18 ATP. Don't forget, you made 2 NAD from transition prep, so that gets you another 6 ATP, and also you have a couple NAD from glycolysis. So that's the whole reason, is to generate as many NADs and FADs to take them to the ETC. All right, and I'm going to teach you a song for this as well. All right, so this Krebs cycle happens twice for each glucose because you made 2 acetyl-CoAs. All right, so let me move myself. What did we put into the citric acid cycle? 2 acetyl-CoAs, some oxidized NADs and FADs, and some ADPs. And what did we get out of it? 4 CO2s, 6 NADs, 2 FADs, and we did substrate level 1 ATP each time. Now, remember, glucose has 6 carbons. We broke it in half into 2 3-carbon molecules, yes? Then we did transition prep. Here's 1 carbon, here's 2 carbons. Then we take this acetyl-CoA, put it through the Krebs cycle, right? And how many CO2s did we lose? 1, 2, right? We take the other acetyl-CoA, put it through the Krebs cycle, 1, 2. All 6 CO2s now, I'm exhaling. For every molecule of glucose that I have, 4 come from the Krebs cycle and 2 come from transition prep. Okay? Now, I am going to teach you a song in just a bit, but I want to show you something here. Okay? Every time we pull off a CO2 and reduce an NAD, in my song I'm going to teach you, that's called do the bit. So this part of it, okay, just teach you a little bit of it. You have a 2 carbon plus a 4 carbon, so we go 2 plus 4 is 6. Okay, look right here. 2 plus 4 is 6. 2 plus 4 is 6. Do the bit, get 5. Now, when we do the bit, we are pulling off a CO2 and reducing an NAD. And then look, we're going to do the bit again. Do the bit, get 4. We're going to make an ATP, reduce an FAD, reduce an NAD. And then we're doing it again because we have another acetyl-CoA. 2 plus 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Okay, so that is part of a song I will teach you, but it also is in the notes. Okay, so let's look at your notes on the citric acid Krebs cycle. For each glucose molecule, the cycle occurs 2 times because you have 2 acetyl-CoAs. In the song, do the bit means remove a carbon as CO2. Okay, you're going to remove a carbon as CO2. You need to put that in there and reduce an NAD. So the products, after 2 turns around, right? 1 acetyl-CoA gets fed through, then the other acetyl-CoA, is you get 2 ATP, because you get 1 a turn. You get 2 CO2, the 2 times we do the bit, right? And since you're doing it twice, that's a total of 4, right? 2 ATP, 4 CO2, a total of 6 reduced NADs, okay? 1, 2, 3, and I do it twice, 6 NADs, 2 FADs, because I do it twice. And then the NADs and the FADs are all going to go to your electron transport chain, okay? And that's where we're headed now. We're going to make some money. All right, let me move and move. Okay, I'm going to go over here. Or over here. Yeah, I'm going to go over here. All right, so let's get our bearings. Remember the mighty mitochondria, and there's that folded inner membrane, okay? This space right here is what's called the intermembrane space. This is the folded cristae, and then this right here is the very center of the mitochondria. That's the matrix, okay? Remember what we learned in chapter 6 about creating a hydrogen ion gradient? So let's see how it's done. Your NADs and FADs have on-ramps to the electron transport chain where literally they take, right, they're our mule carrying hydrogens and electrons. They're going to drop these high energy molecules off, and powerful, strong NADs get to come to the very tippy top of the chain right here. This is their on-ramp. They drop off their electrons, which will get passed through the chain. Remember that analogy we had with the stairs, right? And so they're going to come in as high energy and they're going to go out as low energy, these electrons. FAD doesn't get to come in at the tippy top. He has less energy. He's a mule of lesser status. So he comes in right about here. Okay. What's the significance of that? Well, if you come in part way, then when your electrons are passed along, you are only able to pump out 2 hydrogen ions. Therefore, when they come back in, you only make 2 ATP. But strong NAD who come in here at the top, they get to pump out 3 hydrogen ions with the power of the electron. 3 hydrogens come back in through ATP synthase, and you make ATP. All right. So where are these hydrogens? In the inner membrane space, the outermost compartment of the mitochondria. Okay. NADs and FADs are in the matrix, dropping their electrons off. They're going na. And at 3 different spots, you're having enough energy. That protein at that spot gets enough energy by holding on to that high energy electron to grab a hydrogen from the matrix and bring it out to the inner membrane space. Okay. As you build this up, they want to come back in. Why? A difference in concentration of hydrogen ions, a difference in charge because they have positive charges, and a difference in pH. This is a low pH. So these are your people outside of Disneyland, and they really want to come in through ATP synthase complex. Every time they come in, you make an ATP. So take a look at your notes for a minute. It says the electron transport chain, and this is the cristae, the inner folds, inner membrane, okay? And its purpose is reduced NADs and FADs from glycolysis, because we made some in glycolysis, from transition, and from the Krebs cycle, 2 + 4 is 6, okay? All of those bring and drop off their electrons to the ETC, okay? Now they're oxidized, right? Now our mules are empty. They're not carrying anything anymore, no more cargo, so they can go back and get reused again. And that's what it says there in your notes. They can become oxidized and return back to the earlier steps of cellular respiration. As the electrons pass down the electron transport chain, going from a high energy to a lower energy, their energy is captured to pump hydrogen ions from the matrix, right, to the inner membrane space, and that is where they are concentrated. That is where they are concentrated, in the inner membrane space. As the hydrogen ions move from the intermembrane space back into the matrix, right, they go through a channel enzyme called ATP synthase complex. And they go into the matrix and ATP is formed. Now, as far as the math, okay, strong NAD come in at the top, FAD come in 1 level lower. Do you remember where glycolysis occurred? Do you remember that it happened in the cytoplasm? Those NAD are some lazy donkeys, okay? Sometimes they don't feel like bringing all of that electron energy all the way into the matrix. So sometimes they hand it off to FAD. So some of those NADs from glycolysis won't get you 3 ATP. They will only get you 2. Again, if there is not the second law of thermodynamics, okay? And that's all labeled right here. So now let's look same. I'm not changing anything, okay? All I'm doing is showing you another picture of the same thing, all right? So here, you can see glucose is getting oxidized and you form NADs from that and from transition prep and from Krebs cycle. Here's NAD that is strong, comes in at the top of this electron transport chain. Electrons are moving down. So the electrons are moving through the chain, and at 3 spots, okay, this right here, number 1 is NADH reductase. So reductase is where 1 hydrogen can go out. Then at Roman numeral 3, cytochrome reductase is another place where a hydrogen can go out. And then here at number 4, cytochrome oxidase is another place where a hydrogen can go out. Okay, and so that's your electron transport chain. Chemiosmosis is when your hydrogens come back in to make ATP. Now here's something super important. This is why it's called oxidative phosphorylation. The electrons have an indoor, but just like you have an indoor and you have an outdoor. If your outdoor got backed up, you'd stop eating, okay? If you couldn't get the food out of your digestive system. We need to pull it out of there, right? I know that was gross. I probably shouldn't use that analogy, my bad. Okay, but electrons go in and electrons got to go out. You know who grabs the electrons at the bottom of the chain right there at cytochrome oxidase? Oxygen. So oxygen can take the electrons and hook up with some hydrogen and form water. If you have no oxygen, then these electrons will there's no way to pull them out. Everything gets backed up. So you will have no electron transport chain, you will have no Krebs cycle, you will have no transition prep. And so the only ATP you will get is from glycolysis, okay? And for you and I, we don't have oxygen, we're gonna be passed out within a couple of minutes. It's that important, right? We can't go more than a couple of minutes without oxygen unless you're like a pearl diver or something, okay? So ATP production, NADH entering at the top of the electron transport chain will generate approximately 3 ATP, except the 2 that come from glycolysis, they generally get 2. Okay, FAD comes in farther down, okay? So as a result, he only gets this guy, the cytochrome reductase and the cytochrome oxidase. So as he comes in a little bit further. Here's it's good because there's a picture of a mitochondria right here. So remember, you're concentrating the hydrogen ions in this dark orange region right here. And the NADs and FADs are all here in this lighter yellow part in the matrix. Okay. The final acceptor is oxygen forming water, and then another oxygen will come along and grab some more electrons and so on and so on. And that's why you have water as a product. Okay. All right. Now, this is the picture in your book. It's kind of blurry, but it's the same idea. You can see the NADs dropping off right here, FADs dropping further down, and you can see your final electron acceptor here is oxygen forming water. These hydrogens concentrate out here in the intermembrane space, and so what do they want to do? They want to come back in so you can make some ATP. So that's a picture that's in your book for a reference too. All right, here's a little bit of math for you. Okay, and it really is a good picture because this shows you in the cytoplasm. So here's glucose, right? Glucose, you break it in half, 10 steps, 10 different enzymes, right? And you end up with 2 pyruvic acids. And along the way, right, you reduce a couple of NAD, they're right here, 2 NAD, and you make 2 ATP. You net out 2. Remember, we spent 2, got 4 back, we net out 2. This is through substrate level phosphorylation right here. These 2 NAD, now, if they go in as strong NAD, then 2 times 3, they would give you 6 ATP at the electron transport chain. If they hand off to FAD, only 2. 2 times 2 is 4. So that's where that comes from. Then here, you're taking your 2 pyruvates and you're converting them to 2 acetyl-CoAs. You get 2 NAD from that process. 2 times 3 is 6, right? Here's your citric acid cycle. And notice here your CO2 is coming off the acetyl-CoA and from the citric acid cycle, so all your carbons are accounted for. You do the citric acid cycle twice, you make 3 NADs a turn, 6 total. 6 times 3 is 18 ATP. Then your FAD, you get 1 a turn, so you do it twice, 2 FAD, 2 times 2 is 4. So that's why you're looking right here at 32 to 34 ATP from the electron transport chain, and then a total of 4 ATP due to substrate level phosphorylation here and also here. So that would give you your total of 36 to 38 ATP. And I'm going to add in a caveat. I am telling you, you get 3 ATP for each strong NAD. Really, you get 2.5 due to the second law of thermodynamics. And I'm telling you, you get 2 ATP for each FAD. In reality, you get about 1.5. Okay, so just keep that in mind. If you ever saw a test question like that, that accounts for the second law of thermodynamics. All right, so that is aerobic respiration. And now let's level up a little bit and remind ourselves that it's not just glucose we eat, right? This looks like some stir fry or something here. So carbohydrates, you know what carbohydrates are built out of. They're built out of sugars, right? So ultimately, whatever carbs are in your stir fry are going to get broken down into sugars, which go through the steps we already went through. Glycolysis to pyruvate, acetyl-CoA, citric acid cycle, electron transport chain, ATP, okay? Proteins, remember, proteins are made out of amino acids. So you break up the amino acids and part can come in as pyruvate, part can come in as acetyl-CoA, and part can come into the citric acid cycle. Now, winner, winner, chicken dinner is about fats, okay? I want to remind you about fats. Remember, it's glycerol and those long fatty acid chains. Maybe they could be 18 carbons long, okay? Each of the fatty acid chains can be snipped into acetyl-CoAs. So if you have 18 carbons and acetyl-CoA has 2, there's 9 acetyl-CoAs per chain. And if you have 3 chains, that would be 27 acetyl-CoAs. What has more calories? Per serving, what has more calories? Carbs or fats? Fats. The reason is they generate so many acetyl-CoAs, you get so much ATP that you get all the ATP you need, so you don't need to keep doing cellular respiration. So what are you going to do with the extra carbs? What are you going to do with the extra fats? Store it as fat. Okay, so that's where that comes from. So on, let's see. Oh, can you jump down in your notes? Jump down in your notes to outside of the mitochondria, fermentation, no oxygen required, but then jump below that where it says metabolism at the very bottom. So a catabolic reaction is degradative, and it tends to be exergonic, degradative. And let me just check the next slide because I can't remember. No. Okay. Sorry. Let's go ahead and finish this. Catabolism is degradative. It tends to be exergonic. And in cellular respiration, besides glucose, you can use proteins. You can use proteins at all 3 steps. Okay, and fats can be used. The glycerol can go in as pyruvate, and then you snip the long chains into acetyl-CoAs, and that's why it has more calories. And then anabolic reactions, like we learned in last chapter, ATP and anabolic reactions, right? You're building. So the ATP produced during catabolic reactions will drive those endergonic reactions. Okay? So now we just have that middle piece there on your notes. We need to talk about fermentation. All right. First off, okay, there are a lot of products that are made by fermentation, and it is an anaerobic process without oxygen, without any oxygen. So things like all the alcohols, beers and wines and cheese and yogurts and soy sauce and bread, that is all made by the process of fermentation. Now, take a look. Let me move myself over here. So here's okay, so here is glucose going to doing glycolysis. Here you have pyruvate. If you had oxygen, okay, you would go into the mighty mitochondria and you would finish out. You would do transition prep, you would do the Krebs cycle, and you would do the electron transport chain, and you get a whole bunch of mitochondria because you have oxygen. But in anaerobic respiration, you don't have any oxygen. So what's the problem? Why do you have to do fermentation? Here's the crux of it, okay? Do you remember we had to spend 2 ATP to get our party started, and we got 4 ATP out of it, right? In the process of forming pyruvate, we also formed 2 reduced NADs. We have a finite amount of mules in our barn to carry electrons for us. So if you keep doing glycolysis, all your NADs will be reduced. And you have to have a way to oxidize them so you can reuse them in glycolysis. Now, how do we oxidize them in aerobic respiration? They drop off their electrons at the top of the ETC. That's how they get oxidized. You don't have an electron transport chain in anaerobic respiration. So this is why you do these steps where you either produce alcohol and CO2 or lactic acid like you and I do, which makes our muscles sore, and that is to oxidize our NAD. So let's take a look at this picture. This is a good one. If maybe I can get out of the way. Okay, so there's your mitochondria right there. I'm going to go stand on the mitochondria. All right, here's your glucose with your 6 carbons. All right, glucose, 6 carbons. Remember, we have to invest 2 ATP to get our party started. Then we can reduce 2 NAD and then make 4 ATP. So that's how we net out 2. And we're done with, we have 2 pyruvates. If we had oxygen, we'd go into the mitochondria. But we don't, so we need to have an alternative, and this is why. We want to keep doing glycolysis. Now, if you're a single-celled organism, glycolysis getting you 2 ATP could be enough. It's not for you and I. We can only do this as backup, but some single-celled organisms, this works for them. So if all their NAD is reduced, pretty soon they won't be able to do this. They won't be able to reduce any more NAD, so they won't be able to make that ATP. So here are 2 options. The option you and I do as backup when we're in oxygen debt, like we're going, I'm working out too hard. Okay, what we will do is we will take these pyruvates and we will reduce them right here in our muscles into lactate or lactic acid, which makes our muscles sore. When we, remember redox, if you reduce something, you oxidize something else. By reducing the pyruvate, we can oxidize the NAD, we can unload the mule, right? the electrons and hydrogens that the reduced NAD was carrying, we give it to pyruvate to form lactate. That way we can do glycolysis again. And that's the sole purpose of fermentation, is to oxidize your NAD so you can do glycolysis again. Other organisms, plants, yeast, what they will do is they will take their two pyruvates, they will pull a CO2 off each one. When yeast do this, what does it do to the bread? It causes the bread to rise, right? And then they will take their two pyruvates and they will reduce it down into alcohol, also oxidizing their NAD, just a different strategy. Now, why doesn't your bread taste like alcohol? Because the oven is hot and it evaporates the alcohol off. So on your notes, on the basics, for anaerobic process, the purpose is to oxidize NAD so it can be reused again in glycolysis. So it can be reused in glycolysis again. There are two options depending on the organism. There's alcoholic fermentation. That's the outer path, is alcoholic fermentation by yeast, which produces alcohol as well as CO2. And there's lactic acid fermentation. That's what you and I do as backup. And that's done by bacteria and fungi and animalia, which produce lactic acid. Now, what's the advantages for this? When do we do anaerobic respiration? If you need a quick burst of energy. If you're a sprinter, you're like, right? You're taking off, right? This is not a long distance runner. This would be a sprinter. If you're doing weightlifting, right? Heavy weight, and you need explosive power, that is an anaerobic activity, all right? So the advantage is, I think I have a slide for this, maybe. Oh, wait, let me show you this. Just a moment. Move, Mrs. Sloan, move. It won't allow me to move. Okay, I'll move here. Perfect. Oh, sorry. Okay, so your input is glucose, right? And your output is lactic acid or alcohol and CO2, and the 2-ATP comes from the glycolysis portion, okay? The benefits for doing fermentation, the advantages, a quick burst of energy for organisms like sprinting or heavy weightlifting, quick burst of energy. And it's also used in food production for like cheese and yogurt, bread rising, alcohol, etc. The disadvantages for that, okay, is that, let me see if I have a disadvantages slide. No, I don't. Okay, the disadvantages is it only produces 2-ATP, which isn't enough for us from glycolysis, and it puts you into oxygen debt, where you're breathing heavy, okay, and you need oxygen to recover from it, because basically what you will do, let me go backwards here, when you have enough oxygen, you will take this lactate and you will oxidize it back into pyruvate. And then if you have the oxygen, then you will take the pyruvate and send it through the mitochondria. So that's what you'll do. Okay, so oxygen debt, breathing heavy, it is the required amount of oxygen necessary in order for us to rid ourselves of this lactate. So we're going to have to pay the price to get it back. We can't keep accruing lactate in our muscles. Because lactate is toxic to the cell, it changes the pH, and we need to oxidize it back into pyruvic acid and glucose, and that happens in your liver. So overview, we'll get there. One more, you're almost done. Okay, your overview here, okay, cellular respiration. Glycolysis itself is an anaerobic process, right? Because you don't need any oxygen for that. And glucose goes to pyruvic acid, and you get a couple of ATP and some reduced NAD, right? They're just not showing it to you on here. Fermentation is anaerobic, no oxygen when you do this. And you're either going to make lactic acid or you're going to make ethanol, okay? If you have oxygen, if oxygen is present, you will do transition prep, the Krebs cycle, and oxidative phosphorylation by using all your reduced NADs and FADs, taking them to the top of the ETC so you can generate some ATP. All right. This is a great little summary I wanted to show you. So here's glucose. Okay. And here you can see it's going into this cell. So this light orange right here is the cytoplasm. And you're doing glycolysis, which forms pyruvate. Now remember, along the way, right, we're going to reduce a couple of NAD. Now, if oxygen is present, you're going to take this pyruvate, okay, if oxygen is present, and you're going to bring that pyruvate into the mighty mitochondria, into the matrix, and you're going to do transition prep and convert the pyruvate to 2-acetyl-CoAs, again, reducing some more NADs. Then you'll feed your acetyl-CoAs into the Krebs citric acid cycle, which is what this is, which cranks out even more NADs and FADs. You gather all the reduced NADs and FADs, right? Take them to the top of your ETC, and along your electron transport chain, you will use that to make ATP. If you do not, okay, and here's the NAD from glycolysis is going over there too. But if you do not have any oxygen, then you're going to have to do fermentation in order to oxidize your NAD so you can reuse it again in glycolysis. And your byproduct for you and I would be lactic acid. All right, this was your catabolic pathways. Remember that slide I was looking for earlier? Down underneath metabolism, you have catabolism and anabolism. So catabolic pathways break molecules down, like cellular respiration is a catabolic reaction, releasing the energy out of it. And anabolic pathways require energy. And that is at the very end of your notes. And that, my friends, is the end. Great job. Review, and I'll teach you the song in class. All right.